Hello students, today we will discuss about the folding of embryo. Now when you will see the trilaminar germ disc, you know that it is a flat disc. Now how to see the flat disc? Now in this image you can see that you are having the amniotic cavity in the upper part, and we have cut the amniotic cavity, which you can see here the cut margin in blue color. Now once you will remove the amniotic cavity, you are now able to see the dorsal surface, because you know that this is your ectoderm, this is your endoderm. In the center you will have the mesoderm, above you will have amniotic cavity, in the lower part you will have yolk sac. So when you will remove this upper part of the cavity, that means this amniotic cavity, you are now seeing it from the above. Now when you will see this area, you are actually seeing the dorsal surface of the disc. Now in this dorsal surface, if you will go from anterior to posterior side or from cranial to caudal end, what are the structures lies in the midline? So first is you will have the part of mesoderm is going to form diaphragm, so it is septum transversum. Then you will have pericardial coelom with the developing heart tube underlying. Then you will have this precaudal plate which is going to form future mouth. Then you will have neural tube with underlying notochord. Then you will have primitive node, primitive streak, cloacal membrane, and most caudally is connecting stalk. Now my dear students, if you will see this arrangement, it is a straight flat disc. Now as I already told you, this septum transversum is going to form your diaphragm. So where is the diaphragm in adult body? This is my diaphragm, which is separating thoracic and abdominal cavity. Now in this disc, this is the most cranial. That means if you will make it straight, you will realize that my diaphragm will come here, and below the diaphragm there is a heart. That means this developing pericardial region, and below that you will have the mouth. So this is not the actual arrangement. In actuality, the diaphragm is not the most cranial, diaphragm is somewhere here. So why it is there? Because this area has to go downward and it has to fold itself, and when this rolling will take place from upper end and lower end, that is known as folding of embryo in midline. In the similar way, this is also showing the folding on the borders or the margins of the disc, and ultimately this disc is no more remain flat, it will become cylindrical. Clear? So now the question comes is, what is the reason of the folding? The folding is initiated by the very high speed growth in the central part of the disc. Now in the center part, there are two things which are initiating the folding. One is this midline structure which is known as neural tube, and there is second is appearance of the somites in paraaxial mesoderm. So this center portion is growing very fast, and this peripheral portion is having very slow growth. So the growth into the central portion plate is very high pace as compared to the peripheral portion, and the neural tube elongates, along with that the somites grows, and because of this very high speed of growth, this plate has to fold near the margins. So here I have to explain first the diagram which we are going to use. Now when you are seeing this diagram, this is your anterior to posterior, that means cranial to caudal orientation. So how you cut? You cut this embryo in a midline, but not in the transversely, longitudinally. So it is a longitudinal section. This is your anterior part where you will have this anterior neuropore, dilated part of your brain. Posteriorly, this is cloacal membrane. So we are cutting it longitudinally into the craniocaudal direction. So that's why when you will see the structure, here you can see this is the septum transversum. This is your coelom of pericardial region with underlying developing heart. This area is your precaudal plate. Then you will have this notochord with overlying neural tube. Posteriorly you will have this primitive streak, then you will have cloacal membrane, and this is your connecting stalk. So my dear students, whenever you are drawing the diagram, you should have the orientation that this orientation is cranial to caudal in the midline. Now we are going to fold the area which is anterior to the neural tube, and we are going to fold area behind the neural tube. So these structures will go downward, and when they will go downward, they are going to form your anterior and posterior folds, which are known as anteriorly head fold and posteriorly tail fold. Now, first we will discuss this area, that is your anterior region. So, this is an enlarged view of the anterior region that we will discuss now. So, what is head fold? Head fold is the fold which is seen in the cranial end, and what is happening actually that near the cranial end the neural tube is enlarges and it projects dorsally into the amniotic cavity. So, you know that there is a amniotic cavity in the upper part, and this upper part where you will have amniotic cavity, the floor is formed by the neural tube. So, this is your amniotic cavity in the upper region, this is the neural tube, and as the enlargement of the tube starts, it starts to project into this amniotic cavity. But simultaneously, the growth is not going to stop, the growth is at very high pace. So, when the growth will taking place into the neural tube, now this anterior part of the tube start to project. Now, this start to project anteriorly, or you can say that it overshoot. Now, which structures are now hidden by this overshooting part? This is your area of precaudal plate which is going to form your oropharyngeal membrane. So, this part overshoot the oropharyngeal membrane, but still the growth is not stopped. Further, it will go more anteriorly, and now it overshoot this developing area of the heart which is known as pericardial region. And finally, because of this much of projection, it pushes this anterior part downward, and when there is a downward rolling occurs, you are having a folding of anterior or cranial part of the flat disc. Clear? So, what are the steps? First, there is a growth of anterior end which is initially project into the dorsally in the amniotic cavity, then it overshoot the oropharyngeal membrane area, then it overshoot the pericardial region, and ultimately the folding will take place. Now, because of the folding, there are three structures which position has been changed. What these are? Septum transversum, primordial area of developing heart with its coelom, and oropharyngeal membrane. So, what is the new position of all these three things? They will reach on the ventral aspect of developing neural tube. Clear? So, now the question comes is, what are the result or effects occurs due to head folding? So, my dear students, the first result is formation of foregut. Now, what is happening here that when this anterior part is going downward and roll out towards ventral aspect, now there is a yolk sac is there in the lower portion of neural tube. Now, this yolk sac which is lined by endoderm, now this area of the yolk sac trapped between the anterior fold and the neural tube. Now, here this trapped area is now known as foregut. So, this foregut is dorsally lined by the developing neural tube, and ventrally now it is lined by the oropharyngeal membrane, pericardial region, and septum transversum. So, this is the first and very important result of the head folding, formation of foregut. Now, foregut lies between the developing brain dorsally and the heart oropharynx ventrally. Clear? Now, the second thing is, the brain is now become the most cranial end of the tube, most cranial end of this embryo. Now, when you will see the unfolded or flat disc, what is the cranial most part? Answer is septum transversum. But after folding, what is the most cranial part? Answer is developing brain. Now, the third thing is, there is a change in the position of septum transversum. Earlier, the most cranial position is septum transversum, but now it lies caudal to the developing heart. Now, my dear students, if you will see these two diagrams, you have to understand that in a flat disc, what is the relation from anterior to posterior? This is the one number, that is your septum transversum. This is the second number, this is your pericardial area, and this is your third number, which is oropharyngeal membrane. Now, once all these three will go ventrally, now this is your septum transversum, which is one number, this is pericardial region, which is 2 number, and this is oropharyngeal membrane, this is 3 number. So, from cranial to caudal, the relation is 1, 2, 3. Here, what is the cranial to caudal? 3, 2, 1. That means this 1 number of septum transversum, which is most cranially, now comes posteriorly. Now, this is the 3 number, which is most posteriorly in the 3, now comes anteriorly in these 3. So, it is 1, 2, 3, and this will become 3, 2, 1. So, the septum transversum position is changed. Now, it is no more cranial most structure after the formation of head folding, and the forebrain or developing brain is the most cranial structure after head folding. Now, the change in the position of primordial heart with its coelom. Now, see, this is very important concept to understand, because when you will see the development of heart, you have to understand that which is dorsal and ventral. So, here now if you will see, this is your dorsal surface of this flat disc. Now, here you can see that there is a formation of coelom, and this coelom divide this mesoderm into the somatopleuric and splanchnopluric. Now, under this there is a splanchnopluric where you will have this developing heart tube. Now, by seeing this flat disc, you are having the idea that coelom is on the dorsal side and the tube is developing on the ventral side, because the tube arises in the splanchnopluric intraembryonic mesoderm. So, tube lies initially below the coelom. But when the folding will take place, you can see that the things become reverse. The coelom will go down, so the coelom will go down, and this tube is now become upper structure. So, the relative relation of the tube with its coelom will change with the formation of head fold. So, what is the change is taking place? That initially the coelom is in the dorsal side, but later on after the folding, the coelom shift to the ventral side of developing heart tube. Then the change in the position of oropharyngeal or buccopharyngeal membrane, which I just explained you that it is now not present behind the heart tube, now it comes before the heart tube. Then there is a very important surface landmark appears, that is known as stomatodeum or stomodium. Now, stomodium is a surface depression, and here you can see that there is a 2 elevations are formed. Now, this is your brain vesicle elevation due to developing forebrain, and this is your pericardial swelling or the prominence because of underlying developing heart. Now, in between these 2 swelling, a depression appears on the surface, and this depression is known as stomodium, and you know that this is the part of amniotic cavity, because this is your amniotic cavity. The floor is lined by the ectoderm, and if you trace this ectoderm is also lining this stomodium. So, what is stomodium? Stomodium is a surface landmark, appear as a depression between the forebrain and the developing heart, and this depression is lined by surface ectoderm. And in the stomodium, you will have the oropharyngeal membrane, which is going to rupture and form your future oral cavity or mouth. Clear? So, what are the changes taking place due to the head folding? One is formation of foregut. Second, the brain is become most cranial part. Third, the position of the septum transversum change. Then the proposition or the relation of the heart tube and the coelom will change. Then the change in the position of oropharyngeal membrane, and lastly there is a formation of stomodium. Now, the next is tail fold. Now, my dear students, in the same way, when you will see the posterior part of the streak, you know what are the structures lies on the dorsal side. The most posterior part is the connecting stalk. Now, in front of the connecting stalk, you will have the area, and this area is known as your cloacal membrane. So, this is your cloacal membrane. Then the next is your primitive streak. So, posteriorly, if you will go behind this developing notochord, you will find 3 areas. What these 3 areas? Posterior most connecting stalk, cloacal membrane, and primitive streak. So, when there is a folding will take place, all these 3 things will rotate and come on the ventral side. So what is the initiation of the formation of tail fold? Again, there is a growth occurs at the caudal end of neural tube, and this growth or enlargement of the caudal end pushes the cloacal membrane ventrally. Now when the cloacal membrane goes ventrally, it is taking along with this, what it is taking along with primitive streak and connecting stalk. So now in the next step you will realize that it is no more remain flat on the posterior side, it also curved ventrally. Now here you are able to see one more important thing, that when there is a formation of connecting stalk, a small portion of the yolk sac projects inside the connecting stalk, and this projection is known as allantois. And you know that in human this allantois is not make any part of the urinary system, but it persists as a urachus after the growth. Clear? So what are the results of the tail folding? The first result is that there is a formation of hindgut. Now what is hindgut? The hindgut is the part of your yolk sac which trap into the tail fold. Clear? So the part of the yolk sac which trapped into the head fold is known as foregut, and part of the yolk sac which trap into the tail fold is known as hindgut. Now the distalmost part of the hindgut later on enlarges, and that is going to form the cloaca. Now the second thing which are changing here is the primitive streak lies most caudally. Now see, this is the again important concept to understand, that initially when there is a flat disc, we have seen the most caudal structure is connecting stalk. But here if you will see the relation, what is the relation is, now this is your primitive streak, this is your cloacal membrane, and this is your connecting stalk. So what is the relation from cranial to caudal in the tail end? 1, 2, 3. Now when the folding is taking place, what will happen here, that where is your connecting stalk? So this is the area of connecting stalk, so this is 3. Now where is this cloacal membrane? So this is the area of cloacal membrane, that is 2. Now where is the primitive streak? So this is your primitive streak, so this is your 1. So what is the relation here? 1, 2, 3, and what is the relation here from anterior to posterior? 3, 2, 1. So here what you are realizing, that primitive streak lies anterior to the cloacal membrane and cloacal membrane lies anterior to the your connecting stalk. But now the connecting stalk lies most anteriorly, then you will have cloacal membrane, and primitive streak is most caudally. So the most caudally what structure lies? Answer is primitive streak. Now change is taking place in the position of connecting stalk also. So now connecting stalk is no more present caudally, now it shifts on the ventral aspect of developing embryo, and along with that not only the connecting stalk is shifting, this allantois is also changed its position, and now it shifts into the ventral aspect. So my dear students, when you will have the orientation of the flat disc, what you are able to understand, that this is your area of neural tube. Now this area itself remain as such, otherwise these 3 structure on the posterior side and these 3 structure on anterior side will shift on the ventral aspect. And because of this, what will happen, this lower cavity, that means yolk sac, decreases in the size, and this overlying cavity, that means the amniotic cavity, increases in the size. Clear? So when you are writing the anterior and posterior folds, you have to keep this thing in mind, that 3 structures of the anterior side and 3 structure of the posterior side moves ventrally. Now the next part is lateral folding. Now what is the cause of lateral folding? I told you that lateral folding occurs basically because of the increase in the width of the somites along with the enlargement of neural tube. Now what will happen, that the lateral folding means the right and lateral folds rolls ventrally. Now what is happening, that the folds roll ventrally, and again what will happen, that when they will roll ventrally, they will enclose the part of the yolk sac between the 2 folds, and that part is going to form your midgut. So What is foregut? The part which entrap into the head fold. What is hindgut? The part of yolk sac trap in the tail fold. And what is midgut? The part of the yolk sac that trap between the right and left lateral folds. Clear? Now, what are the effects of the lateral folding? So, students, the first thing is that the disc is no more flat, it has been changed its shape and it is now cylindrical in shape. The second thing is that I just explained you there is a formation of midgut. The third effect is that there is a formation of a important structure is known as vitello-intestinal duct or omphaloenteric duct. Now, what is this omphaloenteric duct? Now, when you will see this diagram, now this diagram is not in the antero-posterior longitudinal, but for seeing the lateral folds, you have to take the transverse section. So, it is a transverse section of your developing embryo. Now, this is your amniotic cavity, this is your yolk sac, and when these two folds are going in the ventral aspect, you are able to understand that these are the formation of the areas of lateral fold. Now, as these folds are going down and down, what is happening that these are enclosing or approaching each other and there is a narrowing occurs into this yolk sac. Now, initially the part of the yolk sac broadly divided into the two parts, but when the folds are become more closer to each other, this communication between the two parts decreases in size and convert into a duct. Now, that duct which is connecting this outer part of yolk sac with inner part of yolk sac is known as vitello-intestinal duct, and this inner part of yolk sac now known as midgut, and this outer part of yolk sac now known as umbilical vesicle. Clear? So, the connection of the midgut with umbilical vesicle is known as omphaloenteric or vitello-intestinal duct. Now, what is the fourth effect? The fourth effect is there is a formation of dorsal mesentery. Now, my dear students, this is very important concept, and dorsal mesentery is arised from, answer is splanchno-pleuric intraembryonic mesoderm. So, please try to see here in this diagram that where is the splanchno-pleuric intraembryonic mesoderm. So, this is your flat disc. You know that on the lateral side there is a formation of a coelom initially, and this coelom divide the lateral plate into the somato and splanchno-pleuric intraembryonic mesoderm. So, now the lateral folding starts, and once the lateral folding starts, you have to concentrate on the two things. One is the coelom and second thing is splanchno-pleuric mesoderm. So, now this is the coelom. You can see the coelom is here. You can see this is the coelom here. Now, where is the splanchno-pleuric? So, splanchno-pleuric is a mesoderm which lies outside the yolk sac. So, this is your yolk sac. So, this is your splanchno-pleuric intraembryonic mesoderm. Now, as the fold increases in the size, here this is your splanchno-pleuric intraembryonic mesoderm, and this is your coelom. Clear? Now, once these two folds comes more closer to each other, now here what is happening? Now, this is the formation of the midgut, and now this splanchno-pleuric of both the side continue with each other, and they are forming a suspension of this posterior part of the gut tube with the wall, and this is known as dorsal mesentery. Now, my dear students, one concept has to be clear here after seeing this formation, that dorsal mesentery is formed by, answer is splanchno-pleuric intraembryonic mesoderm. Now, the second question is visceral peritoneum is formed by, answer is same splanchno-pleuric intraembryonic mesoderm. Because so many times when you are reading the abdomen in your abdomen part of gross anatomy, this question asks so many times that the mesentery of intestine develops from, or it is a bilayered structure of visceral peritoneum or parietal peritoneum. Now students, keep this concept in your mind that somatopleuric give rise to the parietal peritoneum. That means this is your somatopleuric and this is your parietal peritoneum. But here we are forming the visceral peritoneum and dorsal mesentery both from your splanchno-pleuric. That means if you have this question that mesentery is a double layer of visceral peritoneum or parietal peritoneum, I think you should reply it is a visceral peritoneum, because visceral peritoneum and mesentery both are arising from splanchno-pleuric. Clear? Now the other thing which you have to monitor here, that this intraembryonic coelom which is enlarging with the every folding is now lies here, and when these two folds permanently join with each other, these right and left coelom also join and they are converting this into a peritoneal cavity. So in these transverse sections of the embryo, there are two things are very clear, that this splanchno-pleuric mesoderm is going to form your dorsal mesentery and visceral peritoneum, and this intraembryonic coelom enlarges and finally it is going to form your peritoneal cavity. So this is the fourth effect, formation of dorsal mesentery, and this is the fifth effect, because of the lateral folding there is a formation of peritoneal cavity. Now what is the sixth effect of the lateral folding? The sixth effect is that there is a shifting of amnioectodermal junction. Now what is amnioectodermal junction? Now for that, if you will see this diagram, what you are able to understand, that when you will have a flat disc, now in the flat disc, where is the amnioectodermal junction? Now this is your amniotic cavity in the upper part, and here you will have the ectoderm, which is the floor of your amniotic cavity. So somewhere here you will have amnioectodermal junction. Now when the folding will take place, the amnioectodermal shifting occurs as a result of the formation of tail fold, head fold as well as the lateral fold. So all the three sides of the folds, when they are converging on the ventral surface, what is happening? This amniotic cavity is enlarging, this yolk sac is decreasing in the size, and this junction of amnioectodermal is shifting ventrally. So when the ventral shifting is taking place, here you can see what will happen. All the layers, all the amnioectodermal junction, all the amnioectodermal shifting occurs around this ring. Now this ring is known as umbilical ring. So what is the shifting of amnioectodermal junction? That this junction of amnion and ectoderm shift to the umbilical ring, and area below the umbilical ring is lined only and only by the amnion, and because you know that this is the part where you will have the formation of your umbilical cord. So what is the now outermost layer of umbilical cord? Answer is amnion, not ectoderm, and this remaining area of the developing embryo is lined by the ectoderm. Clear? So because you can see here what is the final output, that our surface, body surface or the dorsal surface is lined by the ectoderm, while there is a formation of umbilical ring, and the area which is outside the umbilical ring is going to form your umbilical cord, which is not lined by the ectoderm, it is outside lined by the amnion. Clear? So my dear students, these are the some effects which are taking place after the lateral folding. So what are these? First, formation of the cylindrical shape, second, formation of midgut, then there is a formation of vitellointestinal duct, then there is a formation of dorsal mesentery from splanchno-pleuric intraembryo mesoderm, formation of peritoneal cavity, and lastly the shifting of amnioectodermal junction around the umbilical ring, so the developing umbilical cord is only lined by the amnion and the body surface lined by the ectoderm. So my dear students, at the end of this class of the folding, you should have the idea about the formation of foregut, midgut and hindgut. Second thing, you should have the idea about the results which are taking place after the formation of head fold, tail fold and lateral folds. So this is all for the session. Thank you.